This morning I finally had enough time to go through them, and I've already found about 25 typos. There's one particular typo I want to call your attention to. I use interchangeably V, the letter V, and nu, the Greek letter nu. V is always only the vibrational quantum number. Nu is for frequency. So once in a while you're going to see I had that wrong. V is always quantum number. Nu is always frequency. And nu bar is a special kind of frequency. So I apologize for that in advance. Okay, I think I'll go ahead and get started. So I'm from Stanford University. This is a picture of our campus. It's a classic Spanish architecture. Beautiful campus, a large campus, located 30 miles south of San Francisco. I'm going to be telling you about molecular spectroscopy and specifically two particular experimental methods, laser absorption and laser-induced fluorescence. This should have been first lecture. I'm just going to talk briefly this morning or this afternoon about some motivation. I'll come back to that. So I apologize. That should say first lecture. If you're not familiar with Stanford, you're familiar with the Golden Gate Bridge, which is 30 miles to the north. So we're located 30 miles south of the Golden Gate Bridge. I graduated from Stanford in aeronautics and then came back to the ME department in 1972, so I've been around there a very long time. People who come there never leave. All right, so in the way of an overview, I want to introduce you to some fundamental ideas about spectroscopy and photophysics. They go together. I'm going to emphasize two particular laser diagnostics because I think they're the most quantitative methods that we have. I'm going to spend 2 days talking about shock tubes as a way that we study combustion chemistry with lasers. And along the way, I'm going to give you lots of examples because I know that your backgrounds are different. But if I give you a range of applications, you're going to maybe be stimulated to see the potential power of laser diagnostics or engineering science. We'll be looking at propulsion flows, engines, shock tubes, a lot of varieties with these different diagnostic methods. Okay, now, what is spectroscopy? It's basically the interaction of light with matter. In our case, we're only talking about gases. And the primary examples might be absorption and emission. If you look to the right here, there's a plot of the fractional transmission, one is at the top, versus frequency and wave numbers. That's 1 over wavelength. This is in the infrared at about 4 microns. It's hydrogen bromide. And what you'll see is there's a pattern of lines. We call these lines. A line is the wavelength or the frequency at which you see a specific molecular transition. So what you'll notice is that that molecule has a very regular pattern. And every molecule is different. That's the power of spectroscopy. But there's some regularity here that is related to the structure of the molecule that we can take advantage of. So you can use spectroscopy to characterize a molecule and its bond lengths and its angles, or you can use that information to calculate the spectrum. So it goes both ways. Now the simplest form of spectroscopy for high temperature gases would be emission. You might look at this. This is from one of my laboratory experiments for my lab class. It's the emission that you see in electrically excited air in a specific wavelength range between about 500 and something up to about 600 nanometers. So if you looked in that region of this low pressure discharge in air, you would see this pattern. So you'd say, well, that's pretty complicated. But if you look closely, the pattern is repeated. It's repeated. So there's something about the spectroscopy that's repeated here. And furthermore, there's some structure within here that we'd be able to see if we had high spectral resolution, which we don't have in most cases, but we do with lasers. I'm going to skip over this plot right here. So the reason we use lasers is that they give us intense power, collimated beams. Often they're monochromatic, so that we can do almost perfect spectroscopy experiments where we can take advantage of the shapes and character and positions of lines. So lasers allow us to do laser-induced fluorescence, Raman spectroscopy, laser-induced incandescence. We might use lasers also for doing particle imaging, velocimetry, a lot of different methods that are enabled by the high spectral intensities of lasers and their collimated nature. So we like these techniques because they can be very sensitive and they can also be very quantitative. And I'm going to focus a lot on quantitative. I'm going to just give you some examples. And these are now drawn from energy sciences. The next page will be propulsion. So there's a lot of example applications. You might be interested in remote sensing of pollutants, the combustion efficiency, the process control, a lot of things that you might care about. The parameters we usually care about are species concentrations, temperature, pressure, density, velocity, mass flux, thrust, so on. So those are the parameters we care about, and we're going to get back by studying spectroscopy. So I'm going to show you a few examples here. This was the first OH PLIF image in a spray flame, 1986. That student's vice president of a company in Boston. PLIF stands for planar laser-induced fluorescence, which was something we developed at Stanford around 1982 or so. But we can also study, so that's a simple laboratory flame, but we can also study something big. This is a coal gasifier, which is complicated by the fact that the pressures are high, 15, 20, 30 atmospheres, high temperatures, particle laden, very challenging environment. Swirl burners, we did that, I think, at a visiting trip to University of Cincinnati, Professor Gutenmark. This was a gas turbine combustor study. We've been to coal-fired power plants. So these devices that I'm talking about are useful in the laboratory, small scale, and they're useful in the field. And we occasionally have this capstone project for the students to take their measurements to the field. And here's some incinerators. John Farrell's not here, but these were from Exxon, a project we did for Exxon on incinerators, petroleum place. So you get a sense of the scale of things. These are what I call energy sciences. Now let's look at propulsion ground test. I like to think that optical diagnostics have overlapping value from three different domains. Sometimes we study things that are where you're doing a very sophisticated ground test facility. This is a scramjet, supposed to say scramjet test facility at Wright-Patterson Air Force Base. Very big, expensive to run. They roughly know how it works, but they need to characterize the facility, and we make use of laser diagnostics. Sometimes you're studying something that is so difficult we don't even understand it yet. And the lasers will be used to try to understand a complex environment. This is a 5-tube pulse detonation engine that we studied out in the desert in China Lake some years back. As one of my students did this in the exit plane of a Titan IV. So these are flow fields which we don't even understand yet. So you're trying to just use in situ imaging or measurements to characterize these difficult flow fields. Other times, the facilities or the flow fields are well understood, and what you're trying to do is generate high quality data that can be used to validate models of this simple scramjet. This is an injected flow of hydrogen and a high speed cross flow of air, and that's an image of temperature. So, or we might be in an IC engine. So lasers are used both for high quality validation of say CFD tools, to try to understand very difficult environments where in situ probes are not useful, or maybe to characterize these facilities. And for example, we've been to one hypersonic shock tunnel, only runs for a millisecond or so, and can we characterize what happens in a short time in a high enthalpy facility? So these ideas are applicable to large-scale systems and also to laboratory science. One of my favorite tools is the shock tube. This is a long tube, 100 diameters long. You break a diaphragm, send a shock wave down, it reflects and leaves behind a static sample of high temperature gas. You can pick any temperature you want. We work down to 600 degrees, we work up to 11,000 degrees. Static sample which we probe with various wavelengths of light in order to measure what's happening inside as a function of time. And we use this for chemistry because it's a nearly ideal environment. It's short duration experiments, sub-millisecond to tens of milliseconds, no heat transfer effects, no wall effects. It's a very clean single pulse experiment though. And it has good clear optical access for quantitative measurements. Okay, now I want to say something about just some example applications in the shock tube. Let me show two. We could study, if you were in Professor Pilling's talk today, you would have seen the same graph. He showed this plot of this, sometimes called the most important elementary reaction in combustion chemistry, this branching reaction. And I'm proud to say that over the last 20 years or so, the blue and the red came from Stanford University, where we aspire to make absorption coefficient and rate coefficient measurements to a few percent. Now that's an example of a fundamental experiment where the target is the rate coefficient. Other times we're trying to characterize a very complicated chemical kinetic mechanism, and we might measure species versus time following shock heating. We might measure several species, which has the effect of trying to constrain detailed mechanisms that might be comprised of 1,000 reactions. How do you adjust the knobs of something that has 1,000 rate coefficients? Very hard to do. So you need higher quality data than have been available before, and we do this in the form of multi-species time histories. Okay, included in your notes is a list of supplemental reading that might be of interest to you. We won't go over that now. Here's our schedule. We're here in lecture 1, an introduction, and then I'm going to talk about a few fundamental ideas. Then today we'll talk about diatomic molecules. Next we'll talk about polyatomic molecules. Then we'll talk about quantitative emission absorption. That's the equation of radiative transfer, the information that's buried in spectral line shapes. Then we'll get into some serious spectroscopy, where we have to introduce what are called the term symbols. Then we'll move to tunable diode lasers as one of our techniques that we use. We'll have that for a couple of days, 2 days on shock tubes, and then 1 full day on laser-induced fluorescence. That's the plan. Okay, now we're starting lecture 1. That was all preliminary, lecture 1. And there'll be 4 elements in this, and the way the handout notes are arranged, for the most part, is that there'll be, these numbers will be there, so you'll see where you are. There's one of my students that works at Exxon now who is running a tunable ring dye laser. Quantum mechanics. So quantum mechanics is at the heart of spectroscopy. But we don't have time to have a course in quantum mechanics. So what I've learned over time is if you'll accept a few results from quantum mechanics, you can go a long ways. You just have to accept a few things. The basic idea in quantum mechanics is that in molecular systems, small-scale systems, essentially everything's quantized. But when you go to these small-scale systems, these quantized energy levels are separated enough that you could begin to see them. So the energy levels of molecules are quantized. They do not have continuous distribution of energy. And only certain combinations are allowed to couple to each other in the form of emission or absorption. So if you're in state A, there's only some states B that you can go to, and vice versa. So we have to accept, if we accept these rules, we can just about skip quantum mechanics. We'll just accept it. Now, this would be what I call a potential energy well for a diatomic molecule. So this is a distribution of potential energy that's associated with the charges. So a diatomic molecule has positive and negative charges. They repel. And so this molecule will vibrate with a variable internuclear spacing. That's this coordinate. And then that's the potential energy associated with the charges. In addition to that, the molecule has internal energy associated with vibration and rotation. And those levels are shown here. So this is our simple picture of a diatomic molecule. It's 2 masses separated by a massless spring. So the variation in distance with time oscillates. And it's also rotating. And we're going to find that if we just think of this as a classical spring mass rotating system, with just 1 or 2 results from quantum mechanics, we get everything we need. That's kind of amazing. All right, so Planck's law. The energy states are quantized, and we'll learn more about that later. What that means is that you have discrete spectra. So if we have a molecule that's in this state, and typically energy is written as a horizontal line, energy is upward. If we have a molecule or atom in this state, it may undergo absorption and go to another state, or it may undergo emission and go to another state. If you can have one, you can have the other. But those are so-called allowed transitions. It may be that this is not allowed to go directly to here. Quantum mechanics would forbid that. So discrete energy states means discrete spectra. Now, Planck's law, and when I can remember, I put a red box around things that are really important. This is just conservation of energy. Delta E is E upper minus E lower. Not so hard. But now that's equal to h nu. So there's a V. That's supposed to be a nu. Whenever it's a frequency, that's Planck's constant h. So this is the equation that you would have heard from physics, the duality of radiation in terms of photons and waves. So this is saying that that delta E is related to this frequency, but you can also think of it as the energy of the photon. So this is the very famous Planck's law, h nu. But you know that C is lambda nu. Again, that should be nu. So I can substitute until delta E can be written as hc nu bar. So when I can remember, I write nu bar for a special kind of frequency. It's 1 over wavelength. It's a measure of energy. So Planck's law we use to relate these energy levels to the wavelengths for which we're going to see absorption or emission. If you're dealing with small molecules, and oftentimes in combustion we're dealing with small molecules except for the fuels, small molecules have a discrete structure that tends to be isolated. We can see the different features. If you have large molecules, for example, a fuel molecule in heptane, Then you begin to see that the absorption and the emission spectra look like they're all blended together. We can't separate them. That's because there's so many energy levels they begin to overlap. So typically discrete or blended depending upon the size of the molecule. You have to be very comfortable with this. Everybody should be comfortable with it. C equals lambda nu. So if you can always go back and forth from lambda nu. All right, the two main ideas that we like to use are emission and absorption. There are other types of spectroscopy, Rayleigh scattering, Raman scattering. We'll talk about those briefly. In absorption, you send in a bright light with the intensity I0, and the units might be watts or watts per square centimeter or watts per square centimeter per unit frequency, and out comes a little bit less. How much less depends upon this path length and the composition. And so if you scan this laser in frequency or wavelength, you would see these isolated features if you're in a region where absorption takes place. And those are the individual lines, which is another word for transitions that we can see. And that's going to be governed by what's called the Beer-Lambert law that we're going to derive along about lecture 5 or 6. And it just says that the ratio I to I0, the ratio, which is called the transmission, is given by an exponential decay law, which may be written in different ways, but this term is called the absorbance. You can think of it in terms of a cross-section and a path length and a number density. So that exponential decay contains the information we might want. If we know L, and if we know the cross-section, we can solve for N, and so on. So that's a key relationship. That's why I put it in the red box. Okay, I want to communicate the idea that spectroscopy, there are really kind of three components of spectroscopy: lines, bands, and systems. And we have to be comfortable with those ideas. Now, we're back to this. When you see this diagram, it's typically for a diatomic molecule. This is the distance between the nuclei. This is the energy stored. It's called E-electric here, but it's the potential energy associated with the charges. Superposed in here are these horizontal lines that correspond to vibrational levels, which are quantized, and some fine-scale rotational energies, which are quantized. And so we think, the simplest way to think about the energy of the molecule, not the kinetic energy, the internal energy, is that it's the sum of the electric energy or electronic energy, vibrational energy, and the rotational. We imagine that they're independent, and that gives us some advantage. So we think it's simply the sum of all three contributions. And so if we look at an individual transition from one state to another, that's a line. The reason it's called a line, you're going to see, I'm going to show you some exposures on film, and the way those experiments were conducted for 50, 100 years is that the horizontal axis was wavelength and the vertical axis was nothing. It was just another way to gather more light. And so it appeared on film as if they were lines. But they were really just single wavelengths for which light was being emitted or absorbed. So that's a line. That's a single transition. On the other hand, if you looked at the multiplicity of lines that might go along with a vibrational transition between V lower and V upper, and you scan across some spectral region, you would see a family of lines. In fact, you might see this pattern, which I showed you earlier. Those groupings of lines, that group of lines is called a band. And that band is comprised of two branches called R and P. So these individual elements are lines. This group of lines is called a branch, and the two branches together is called a band. A band means the specific upper and lower vibrational states. So these would be the delta V, V upper minus V lower of 1. These are the allowed so-called rho vibrational. So rho for rotation, V vibrational. So rho vibrational means combined rotational and vibrational quantum number changes because they go together. This is for delta V of 1, which is the strongest case, but it's also possible for delta V of 2 or 3 to occur. They're usually quite a bit weaker. So now if I showed you an expanded region of spectral space, here's the delta V of 1 transitions, here's the delta V of 2s, and here's the delta V of 3s. Each time you go from here to here, the intensities go down by about a factor of 100. So these are the strongest ones. This is for CO. So these are called, this is the so-called fundamental, first overtone, second overtone. So these are, each one of these groupings is a band. So bands have lines. Bands have two branches. Now we have to recognize that you can also have transitions that go between different electronic states. So in nitrogen, molecular nitrogen, there will be different electronic states, and they're called A, B, and C, for example. Not shown is the one called X. If you have transitions between these different manifolds of states, all of the lines between these two manifolds is called the system. So the system is like the big picture of all the lines that go from B to A. A different system between A and C. All the lines between B and A is the system. The system would be comprised of many bands, and the bands would be comprised of 2 or 3 branches, and the branches are comprised of lines. Spectroscopy is accounting, just accounting. Okay, now back to this experiment that they do in my lab course. This is the spectrum that we showed earlier. Now I've got some notation on here. This is just arbitrary intensity. So if we look at this low pressure discharge, we scan our monochromator, we record the intensity, and then with a little help, the students understand that this particular transition is a so-called 6-2. It's associated with a transition between here and here, but they're grouped. And the first number corresponds to the vibrational quantum number up here, and the second one is the lower one. So this is V upper and V lower. So 6 to 2, 7 to 3, 8 to 4. You'll notice that all the numbers differ by 4. So this is the group of the delta V of 4 bands. It's also possible to have delta V of 3 or 2 or 1. But these are the primary delta V of 4. So now you're realizing that when you go to systems, there can be a lot of lines. If you have just a band, it's not so. We could blow this up and we could figure it all out if we had enough spectral resolution. So there's a lot of rules being followed here, and there's a lot of notation. But in the end, it's really just lines are in bands or in systems, and you have to decide what you want to study. If we looked at a few different electric discharges in air, I think maybe it's maybe it's wet air, you begin to see a lot of different features in the region between 250 nanometers and, your eyes see between around 400 nanometers and say 750, something like that. That's where your visible is. To the left of that, it would be the UV, and to the right of that is the near infrared. And this is another case of different kind of plasma. So the thing about a plasma is you can have a cheap, low pressure discharge, you get a lot of light out. So you use emission when something is hot. You tend to use absorption when things are cold. The emission comes from excited states, and so all you can do is learn something about the excited states. In absorption, you learn something about the lower states. But there's patterns. And with time, you can figure out these patterns. So which systems are involved, which vibrational bands, and so on. So we look at a single line, and now we start adding them up, and there's a lot of lines here. You might think that they all overlap, but they don't. Okay, this is where the word line comes from, I think. So in the old days, people would use film. Maybe they still use film. People would use film, and they would put it at the exit of a machine called a spectrometer. Actually, they called it a spectrograph. Spectrograph implies that you're going to measure at multiple wavelengths simultaneously and record on something like film, or maybe nowadays a CCD array. So you take light, send it into the box. The box disperses the light so that light at any one wavelength goes to a specific location. So this from left to right is just a distribution of intensity of wavelength. And at any one wavelength, there's just a line that appears that corresponds to a specific transition. And over time, the spectroscopist, the physicist, would learn, well, this is the spectrum of OH for a particular system. That's the 2 sigma to 2 pi system. You'll learn about that later. And for the 0-0 band. So they can figure out what that is. Once it's known forever. OH doesn't change. CH, so on, NH. And so each of these molecules has its own spectrum. You can change the spectrum if you change the temperature, because you populate different states, and therefore different states emit. So by changing the temperature of the emitters or changing the temperature of the absorbers, you can probe different parts of the spectrum and learn more and more about the molecules. So you can use this to study the molecules and say, well, H2O, what's the bending angle of H2O? You can figure that out. What's the internuclear spacing? You can figure that out. So you can figure out a lot from the spectroscopy, or you can use that information to calculate the spectroscopy. So it used to be that people would use film. Now they probably would use CCD arrays. Now we use lasers. And I'm going to talk about absorption and emission. Absorption is a favorite of mine. We send in some light from a wavelength tunable laser, sends through some sort of a medium, might be a flame, onto a detector. The detector just sees photons. How many are coming in? And now, since I told you that the energy states are quantized, then the difference between two numbers is a number, and that line should have only a single wavelength. But then quantum mechanics tells us one more thing, and there's something called the uncertainty parameter, uncertainty effect, which says that when you have microscale systems, if you watch it for only a limited amount of time, there's a limitation on how well you can specify the energy. That's the Heisenberg uncertainty principle. So it turns out that for these reasons, lines actually are not infinitely narrow. They're not a Kronecker delta. They actually have some width. And that's very interesting because these width depends upon environmental factors. So if you were to study the shape of an absorption line, here's the transmission, which I can invert by Beer's law to plot as absorbance. It has some width. Even if I measure this with a monochromatic laser, it'll have width. And that width is a function of pressure and temperature. So all of a sudden, with the laser, we have the ability to use line shapes as a way to measure pressure and temperature. That wasn't possible before we had monochromatic sources. So just to reiterate, there are three key elements of spectroscopy: positions of lines, strengths of lines, shapes of lines. Positions of lines can be determined once we have formula for the allowed energy levels. Once you have formulas for the energy levels and the rules from quantum mechanics that tell you which ones can talk to each other, you've just calculated the positions of all the lines that are possible. But you don't know how strong they are. The strength of the lines, that's something else. That's a different kind of part of science. And so we have to deal with the fact that we need to find out where they are, how strong they are, and then we have to worry about how they're shaped. And those are the three key elements that we need to understand something about to do quantitative spectroscopy. Now we have to deal with what's something called the Boltzmann distribution. And so now I ask the question, how many people in here have not had statistical mechanics? Everybody's had statistical mechanics. Everybody knows S equals K log omega. Everybody's heard this. Okay. So this is really an important thing. This is the statistical mechanics is so important to us because it tells us the fractional population in a given state. If we have a state N1 of a specific molecule or a species, we can calculate the fractional population of that species that are in this state as a function of this very simple relationship. G exponent e to the minus energy over kT. So energy is just the energy from any reference you want to pick. You just pick a reference. But you've got to pick the same reference in the partition function down here, which is the sum over states. So E is the energy of the state, Q is the total partition function, and now notice, if the internal energies are summed over rotation, vibration, and electronic, I can separate those terms and I can write this as the product of three partition functions. That's why we do this. So I can think of this total partition function as the product of individual ones. What's G? To any of you who've had statistical mechanics? Degeneracy. What does it mean? It's the number of states that have the same energy level. So it's because we can't distinguish them. And so if we have an experiment where we can't distinguish them, then we have to count them this way. So Q is the total partition function, G is the degeneracy, and this is just the energy. Now, this is really the definition of temperature. If you tell me temperature, I can tell you the distribution if they're in equilibrium. But if I measure the populations, I can solve for temperatures. This is really the definition, and it's possible to have different temperatures. You can have a rotational temperature that's different than a vibrational temperature. You can have situations that have no temperature. If you have a laser with a population inversion, what's the temperature? Negative. Pretty amazing. So you have to be careful what temperature means. It's your friend, but it can be tricky. But if we have a well-defined temperature, this is how we get the population in that state. Why do we care? Because the simplest concept for absorption and emission is going to be that that signal is going to be proportional to the number in that state. So we need to be able to count the number in that state in order to estimate the strength. Okay, now that's 29 minutes. Okay, that's enough theory for today. What I want to do now is give you a bunch of examples And the idea is just to show you how powerful these ideas are and how far you can go. And all my students are mechanical engineers. They start coming in. They've never had a course in spectroscopy. I've never had a course in spectroscopy. So you learn. But you can learn this stuff. All right, so I've had a vision for quite a long time of using tunable diode laser absorption in aero propulsion. And so here's a kind of a scramjet cartoon. And the idea is that you would have a family of different lasers, all little semiconductor lasers that are, you know, very, very small, each one with a different color or wavelength. Each one maybe can be tuned over some narrow wavelength. And you propagate those things for fiber optics, and you probe different places in your combustor. This is a scramjet combustor, but it could be some other kind of combustor. It could be an internal combustion engine. And each one of these colors is designed to tell you something different. Maybe it's a different species. Maybe it's to give you temperature or velocity, something. So we've learned over 20 years or so, since we've been doing this with room temperature lasers. Did it earlier with liquid cooled lasers. You can probe, say, the inlet or the combustor or the exhaust. And for example, at the exhaust, you might be interested in the temperature, the species, the unburned hydrocarbons, the velocity. You might want to measure the thrust, which is really the product of the mass and the density, rho u squared, basically. Or you could do something up here at the inlet. You can measure the mass flux. We've done this in the inlet of aircraft, on the ground. And the Air Force is trying to do something like this now with some of their scramjet tests. So this is the idea. You would take kind of this, it's the same physics, but you have to know the spectroscopy well enough to pick these colors and to design your experiment. But you can measure a lot of different quantities that could go in this experiment. Over time, we've worked on techniques for temperature, velocity, water, CO2, O2, and others. Generally, we develop the ideas in some simple laboratory system, and then we might take them somewhere on the road. We've been to ATK on Long Island. We've been to University of Virginia, Wright-Patterson Air Force Base, China Lake, a lot of different places. And as I said, the Air Force is now starting to use these ideas in some flight tests. And I think eventually these ideas could be used for control. Right now it's more like what can we measure, what's happening inside, fundamental experiments, but eventually I think we could use these for control. Here's another example. Now that was kind of a scramjet example. This is now a high temperature example. This is, we live nearby NASA Ames. NASA Ames Research Center is about 10 miles away. One of the things they're famous for is they have some high-powered arc jets. They have, I think, a 20 megawatt and a 60 megawatt arc jets. So they heat air at high pressure, expand it in a nozzle, and flow it at high velocities over, say, some tiles that they're testing for the space shuttle. So it's a way to produce very high enthalpy gas, usually air, or at least some oxygen. So you add energy in the form of this constricted arc, and you expand it in a converging diverging nozzle, and you produce the high velocity, low pressure flow that you might associate with hypersonics. It's a difficult experiment because of all the electromagnetic radiation and noise. They put all this in a big box. All those orange lines are water cooling lines. Let's see, I don't know. Well, it's up to 60 megawatts, so there's a lot of water cooling is required to keep this thing alive. It runs for minutes. This is the design. So you have some sort of an anode and then a stack of copper discs that comprise this constrictor tube that might be several feet long. So the arc strikes and goes this to the cathode. And the idea is that you constrict it so that hopefully over that length you heat the gas to some controlled high temperature. And then you, somewhere in here you may add some more gases. And we'd like to measure right here to get a measurement of the stagnation conditions before they expand. So how would you calculate the flow field? You might do some sort of energy balance, you know, how many megawatts are in and how much cooling water's out, and so on, what the flow rate of gases is. But you'd really like to measure it right there. And so we've been working on this for a while, successfully, and then back again. Now the temperatures are going to be in the range of 6,000 Kelvin. It's going to be, say, air at maybe 6 atmospheres. So at 6,000 Kelvin, 6 atmospheres, you can argue that that air will be in chemical equilibrium, because the time scales for reactions are going to be much shorter than the flow time in this channel. So one of the things you have going for you is you know you're putting in air, or maybe air and some known amount of argon. You can assume chemical equilibrium. You can assume that there's one temperature for all the states that you're worried about. And we're going to take advantage of that. And one of the questions we're going to have is whether this flow is uniform or not. So this is a difficult situation. It's high temperatures, high pressures, high currents, so on. Lots of mechanical, optical, electrical difficulties. Now here's the physics. Let's see, I think I may not have time. Okay, optical axis is a problem because it's just like a web of water cooling lines. How do you get light into this duct that's some 5, 6, 7 centimeters across? Optical fibers. So you come in with optical fibers and some special lenses on the end, and you beam it across and pick up the light. That's what we do. This is the energy level diagram for atomic oxygen. It's a little different than the molecular ones I was showing you. So this would be the lowest energy state for atomic oxygen. Here are the next excited ones. At some point we're going to talk about this notation. It's called term symbols. Well, these wavelengths are so short you can't really propagate that light. Even if you had a laser, you can't propagate that light through the air because it's absorbed. That's the so-called vacuum ultraviolet region. But we can excite and detect here. If we use a wavelength of 777, that promotes the absorption from here to here. And now using Boltzmann's relation, I can relate this population to the total oxygen population. And I can argue that the air is in equilibrium. And so if I measure, therefore I know the mole fraction of total amount of oxygen that's present. And in most cases, by the way, essentially the oxygen will be fully dissociated. But in any case, if I make a measurement of the density of this in this one level here, I actually can tell you the temperature because of the chemical equilibrium argument. And so what we do is we scan across this line. Here's the absorption trace. We scan across the absorption feature. There's an individual. Yeah, you'd like to think that that's an individual line. Well, it turns out it's a little more complicated than that. There's some so-called fine structure to this line. There's some sublines. But it adds up to essentially one line. And the integrated area under that feature is proportional to the number density of this. Well, it's proportional to the path length, number density, path length, and cross-section. So, but by integrating over this and knowing the length, we can convert this immediately to the number of atoms per cubic centimeter in that state. And that's enough, together with our knowledge that we started with air, to calculate the temperature rather well. That's the idea. I'll put aside some of these difficulties here. So this is very temperature sensitive. So this is our example. This is the plot of the temperature that we infer, and that's somehow we're missing. This is all in kelvins versus time in seconds. So they ran up to 800 seconds. Here's some starting transients which we can measure. Now it just sits there, kind of hums along at about 6750 Kelvin. So there's no thermocouple here. This is the power of using photons. At this point right here, they, I think they, yes, somewhere in here the last 200 seconds they changed the arc current. And if you look close, you can see that we could actually detect that. They decreased the arc current by about 100 amps out of the 2000. And we could actually see it. So this was working amazingly well. Now what is this? This is a path average temperature. We only have two numbers. We only have I over I0. We scan it, so we really get the path integral of the number density in that state. So only on the assumption of uniformity can we infer this temperature. Or if you tell me the distribution shape, I can tell you the peak temperature. So what I'm not going to show you, I believe, is that we've measured at different locations, different transverse directions to sort out this non-uniformity problem. It's not quite symmetric, we've discovered, which is an important finding. If it's not symmetric, then when it goes through the nozzle and goes over the test article, then perhaps you're going to have non-uniform flow, and that's the thing that they're worried about. Okay, I'm going to go to another example. Looks like I'm maybe going too fast today. For those of you interested in engines, this is a detonation. This is a pulse detonation combustor. It's essentially a pulse detonation engine. Sometimes we call it pulse detonation combustor. And so to the left somewhere is some sort of a system, a pipe, a tube, in which detonations are induced from time to time, maybe 20 hertz. So there's a pulsed flow coming from the left driven by detonations. So you fill the tube with the detonable mixture, set it off, detonation wave comes down, the gas flows out through the nozzle, and then some people have argued that you could take that exhaust and you might be able to connect it to a gas turbine. So instead of having steady flow coming in, you're going to have pulses of flow. That's going to be a problem in terms of survivability of the turbine, but why would you care? Well, you can, this detonation combustor, which might be a meter long, the detonation wave moves so fast that it burns almost like a constant volume. And you can show from thermodynamics that that constant volume combustion can be more efficient than constant pressure combustion normally used in a gas turbine. So there's some thermodynamic potential if you can learn to live with the downside of detonations. Well, so they'd really like to know what's coming out as a function of time and what's going into this downstream unit, which would be some sort of a turbine. So basically you have to imagine oscillating conditions. And you want to kind of know how efficient is it, how much energy flux. So we have a gas dynamics argument here. I think it's here, yes. So inside this test section is a throat. And to the left, it's essentially always choked. So what do we know? We have choked flow, that we know we have sonic flow, and we take advantage of that knowledge together with some measurements at different wavelengths and colors to try to say what's coming out as a function of time. What is the velocity? It's sonic. So if we measure the temperature, we know the speed. We measure the composition, we measure the temperature. So the idea is that if we measure here at the throat, we can sort out enthalpy versus, we can sort out everything, assuming uniform. That's the idea. So we might work with CO2 at this is 2.7 microns. That's in the infrared. CO is in the infrared. We do a lot of work in the infrared. Those are low vibrational transitions. This is the conceptual thing here. So here's some sort of a detonation tube. There's to the left would be some sort of an igniter. So the air maybe is flowing and the fuel comes on, it ignites the detonation wave, and then the fuel comes on again, and that might happen at 20 hertz. The higher the repetition rate, the higher the power coming out. And we'd like to measure what's out here, but we'll do it by measuring in this throat. That's the idea. It's something we've done in the last year or so. So let's see, one student was busy with measuring temperature in water, mole fraction, by looking at the ratio of he measured the absorption in two lines. If he takes the ratio of the absorption in two lines, he's measured the relative populations in two states, and therefore with the Boltzmann relation, he's measured temperature. So he measures temperature in water, and the other student was measuring concentrations of CO and CO2. I think the fuel was usually ethylene. So the main thing is this is a variable pressure, temperature, velocity, time problem. You have to have very fast diagnostics. So, but the key was this idea that if we had choked flow, sonic there, we could sort everything out. That's why we have the red box here again. Time-resolved measurements, the idea would be to challenge the CFD. There's actually not a CFD code for this. It's a little too complicated, but they're trying to develop. This would be a measurement for a single cycle. Let's see, the blue is the temperature measurement, and the red is the blue is the temperature model. The red is the temperature measurement. So note the time scale. Everything is kind of over in 1 millisecond. Then there's some, if you saw, those of you who saw Thierry Poinceau's talk today, showed that nice detonation experiment and the stuff sloshing around. That's basically what happens over milliseconds. The gas just kind of, it blows down and then it kind of sloshes around a little bit there. But notice that we're measuring temperatures up here at 3,500 degrees Kelvin. There's some big differences between the measurements and the code. They're still working on the code because it's a challenging problem. So it was a challenge to get a sensor to work at these are extreme conditions. So you have to understand spectroscopy. in order to design the system that works over. This was only like maybe a 1.5 centimeter gap. I mean, this is a pretty narrow path to work with. But anyway, we figured out ways to work at high pressures and high temperatures. And at least at the early part, it erased pretty well, really. Now let's see. Okay, so once you have the velocity at the throat, which is a function of the mixture and the temperature, so we measure the temperature, we get the velocity, we know something about the mixture, we get the velocity, we can get the mass flux, we can also get the enthalpy flux. And so this would be a plot of the mass flux versus time, kilograms per second here. So it's a very transient problem. The detonation wave comes and drives this velocity way up, and then it goes through some little cycles here. And if you look longer and longer, you see some kind of forward-backward stuff going on. It actually reverses sometimes. Now, from these components, we can get the enthalpy flow rate, which is kind of, in fact, that was what we got the money for. Could we develop an enthalpy flux probe? That was the idea. So we figured out how to do that. So this is a plot of enthalpy flux up to, I know, peaks out here about 14 megawatts. So the idea is, could you measure this? And first thing we see is that it's pretty much the same from cycle to cycle. So that's good reproducibility. And then, of course, you'd look at some sort of integral under this curve to try to get the total per cycle. So I don't think anybody's ever done this before. The idea of measuring highly transient power, mass flow, power flow, integrated information. Now, I'm going way too fast. I'm finishing 14 minutes early. We're going to stop soon. So this was all motivational, introductory, lots of examples to show you why we care about this stuff. Then we transition now and we start talking about molecules. What's the simplest molecule? Diatomic. We had at Stanford, we have a famous professor, Shallow, got the Nobel Prize in physics from the laser wave. He was an atom guy. He hated molecules. He had some funny saying about even a diatomic molecule had one too many atoms. But for us, the diatomic molecule is the simplest molecule, and we can learn a lot from that. It gets a lot more difficult quickly. So we're going to separate the motions. We got well, all the atoms and molecules zip around through space according to a Maxwellian velocity distribution. That's their kinetic energy. We're talking here about the internal energy of rotation and vibration and the spectra that you can get from those energies. We're going to talk about that in some detail for 2 lectures. Now, the first one is diatomic, and then I think the third lecture today is polyatomic. Everything gets much more complicated fast when you go from diatomic to polyatomic. But we'll do that today, and then, sorry, I can't think ahead to lecture 4, but I know what I'm doing today. All right, so I'm going to stop there, and then I'll take some questions, and we won't resume until about maybe 10 after or so. Okay, question?
On slide 29, which was the atomic oxygen absorption,
yeah.
Why is the center of the peak not at the absorption band?
Is it 29?
Yeah, or at least what you have in the book.
Yeah, the book might be slightly different. Okay.
Yeah, that one.
Now, what
so
about it?
The transition is 777.2, but the peak isn't.
Well, you have to recognize that this line is actually the superposition of some fine-scale structure. And you have to understand that and the relative strengths. Although it's symmetric, so I'm still not sure I understand your question.
Well,
I think it should be.
On the left, it shows the transition is 777.2.
Uh-huh.
And on the right, it doesn't look like the center of the peak is at 777.
That's close enough for university work. I hope all the questions are that easy. No, that's just rounding it off. If the students come back and tell me way too many significant figures, I always pound back on them. You know, if you don't have any evidence to support 6 significant figures, then don't give them to me. So this is probably a case where I just told him to keep it simple, 777.2. But we have to be very careful about the actual spacings of these subfeatures. Sometimes you have to worry about subtle things like there may be isotopes. They have slightly different spectroscopy. So there's some kind of hidden subtleties here that I'm not going to get into today. But this single feature is really the superposition of some fine structure. Yes?
For this one, you assume the total number of moles of oxygen, right? Oh, to find the temperature.
Well, you put in air.
Yeah.
You put in air, and let's just take the simple case. Temperature is high enough, all of the O2 becomes O. And now, but you know from chemical equilibrium arguments that if I give you the temperature and the pressure and the relative numbers of nitrogen and oxygen atoms, you can tell me the composition. So let's just say it's at 6 atmospheres. Turn the problem around. It's at 6 atmospheres, and I've told you how many O atoms there are. You can tell me the temperature. So it's just an equilibrium argument that we use. Yes?
What determines how many branches do we have in the band?
Ah, good question. So in the simplest case, it's 2. And we're going to, I don't know if I go that far in this lecture. And you'll notice that they're called P and R. It follows the alphabet. There will be an alphabet soup that goes O, P, Q, R, S. And delta J of 0 is set at Q. I showed you a P and an R, which means that delta J, you're going to learn, can be either plus or minus 1. But there's some molecules where it can be 0, and then we'll see a P, Q, and R. And if you look really close, you'll see sometimes very wimpy O and S's. So what determines it is the character of the molecule. You're going to see it all depends upon the term symbols that we're going to define a little bit later. The term symbols will tell you what you can see. But in the simplest case, like say carbon monoxide, you'll just have a P and an R. That's the simplest case. The next level up is you'll see a Q, and they'll all be piled together, and you'll see 3 branches. And if you look really close, we're going to do a case example with OH. If you look really close, you'll see what are called satellite bands. You can see some wimpy others. But for the most part, it's dominated by, it's either 2 or 3, depending on the complexity of the molecule. Spectroscopy is notation and rules. It's a lot like accounting. Any other questions? Okay, so I'll have to go a little slower next time, I guess. We'll take a 20-minute break and start at, say, 10 after. Okay.